Okay, so thank you very much for inviting me to chat with you all today. And today I'm going to talk a little bit about lots of things. First of all, as David said, we're based at Swansea University here in Wales. This is the picture. Actually, it is quite sunny outside the window today, so this isn't too far from the truth. So we're based here in the Park campus at Swansea University, and the research group that I lead is called SHOAL. So it stands for Sociality, Heterogeneity, Organization, and Leadership. And that's what we're going to start off with today is an overview of the fundamental research and methods that we use in the research group, sort of what you would call the blue skies research that we do, the question-driven, hypothesis-driven research, which is what gets me up in the morning, the thing I get excited about. And then we'll spend a little bit of time thinking about how the work that we do in those areas can then lead on to opportunities to tackle societal and environmental challenges. So how the kind of the pure research that we can do can inform or act as a springboard towards more applied research in various areas. And then I will end today with a little discussion of some of where I think the research field of collective behavior can move forward in the future, and something that me and my group are definitely really interested in looking at and taking a more comparative research perspective. So that's what we're going to go through. I'm told we have up to an hour, and so just jot down questions and make sure you keep them for the end. So to start with the overview of the research, the first thing that we're really interested in is studying sociality. So here you can see a picture of me studying pairs of chacma baboons within a baboon troop. And these baboons are grooming one another, and I'm taking observations by direct observations, and I'm noting down, in this case, focal sampling of what they're doing, who they're grooming, for how long, and all of these behaviors they're performing. And this is in a site in this yellow flag here in Namibia, which is part of the Saubis Baboon Project run by Guy Cowlishaw, and now also with Elise Houchard and AJ Carter. So this is where I did my PhD, and so that was 2003, 4, 5, so 20 odd years ago. And since then, we've developed projects in various parts of Southern Africa, mostly working with Justin O'Rain, who's in the University of Cape Town. So these population down in the southern part of South Africa, and also these northern parts here, also with Cape Town, and this little purple one with Durham University and Russell Hill. So we study baboons in all these different populations to try and understand how and why they are social. And from these direct observations, like I'm making here, we can create networks of who's grooming who. So here we got the nodes representing each individual baboon, the links between them representing a stronger than average grooming bond. And so those which have more strong bonds end up with these thicker lines. So these are the numbers here represent the dominance of these individuals. So you see more dominant baboons end up central in these networks. And in this population, we showed that the grooming relationships that the baboons have with each other predicted the likelihood that you see them co-feeding with each other, so sharing the same foraging patch. So this is a form of tolerance or food sharing in the baboons. Not active food sharing. There's still lots of displacements and aggression, but the grooming could be seen if you're looking at a biological market theory type approach, that you're exchanging grooming for the opportunities to be tolerated at the food patches. So the baboons are happy to co-feed with their close friends. And when we're looking at the baboons like this, you only get to watch a couple of baboons at a time. And if you're looking at social behavior or group behavior, that's quite challenging because you can't see much of the group at the same time. So for a while now, we've been developing tracking collars. Here's Gail Fellman, a former PhD student, putting together some of these collars. And if you're interested in building something similar, all of the protocols and the kit is all online, so you can try and have a go yourself. And when we're doing this, we always have in mind specific questions or hypotheses that we want to test with these specific data which we're collecting. And so what we do is we trap the baboons using cage trapping. The baboons go in to get some food. We set the traps, and then we put them under anesthetic, and then we fit the collars. And this gives us back some beautiful data. So the collars collect acceleration data, which is the three squiggly lines you're seeing here. And this is a visualization suite that we have in Swansea, which is Rory Wilson put together for us. And what I'm looking at is the acceleration signal in three axes, the X, Y, and the Z of this baboon's activity. And these squiggly lines can then be used if you take the acceleration data and take videos of the subjects which you're watching. We can time match that acceleration data to the video data, and we have these labeled data sets. And we use this to train models, random forest models, that gives us predictions of them. There's some seagulls outside. Sorry if you can hear all that squawking. It gives us a training data set, and then we build the model to try and predict the behaviors that we've seen in the videos in the video which we haven't labeled. And that works really, really well. So we can get really high precision and accuracy of predicting what a baboon is doing for every second that they're wearing the collars which we attach them. And most recently, Charlotte Christensen, who's finished her PhD, is now in Zurich as a postdoc, working with myself and Ines Furtbauer, was able to take this on, not only find foraging or walking and running with high accuracy, but also be able to determine grooming amongst baboons from accelerometers in the collars around the baboon's neck. So that means that we are moving away from having these rates of behaviors and these rates of grooming to absolute values of total durations of grooming amongst pairs of individuals for the time that they've got the collars on. And then we can move from this sort of rates and snapshots types of behavior towards different sorts of questions. For example, we could look at the total grooming duration for the baboons. And with Ines Furtbauer and Charlotte, what they did was look at the fecal glucocorticoids in the baboon's poo, and then matched this to total grooming duration. And what we see over a long period is that the more grooming baboons do, the lower these glucocorticoids. So that's what we standard report in the literature. So stress levels are lower the more grooming that a baboon does. But actually, when you look at the total grooming duration, it seems that as a short-term cost, so the more grooming that's happening, the higher the glucocorticoids, which is kind of a counterintuitive result. So by changing the way that we're able to collect these data, we can get insights into different scales of sociality and the causes and consequences. Thinking about baboons, it's really easy to think of, well, they're different. They have different ages, sexes, dominance hierarchies, reproductive status. If you're looking at a shoal of fish or a group of, or a flock of birds or something, perhaps it's a little less obvious how these individuals are different to each other. And you can look at these behavioral differences. You can look at population mean or a group mean. But when you look into it, almost every social animal that's been studied shows these inter-individual differences, and these are consistent over time and context. So, for example, these stickleback fish, these are two females. The video is speeded up a little bit, so they're not quite that fast. And you'll see the fish in the bottom here. She's moving around. They're moving around and exploring that environment. And you probably haven't seen the top one, which is just hidden behind that artificial plant there. And if you test these two fish a month later or 4 months later, the bottom one will still be more exploratory and the top one more sedentary. So these consistent individual differences in behaviors, or this heterogeneity, this individual heterogeneity, I think is fascinating from the perspective of group behavior. And when we have moved on, and this is Francesca Occhiuto, her PhD is looking at dairy calves, and we can use these location sensors and have many, many more individuals, and we can do this over much more bigger timescales. And here we're just looking at total distance traveled in a day, for example. And again, you can see these consistent inter-individual differences in their movement behaviors over the day. And so we can start seeing what the consequences of these differences. For example, with the fish, you can look at total distance traveled, just like we did with the dairy calves there. You can look at space use, time in free water, time near objects. But you also zoom in and look at more metrics that movement ecologists might be more familiar with, like the step length, so the distance they move per unit of time, or the angle which they turn per unit time, and how often they're moving quickly or how often their time is stationary. And you can measure all these types of things, and often they're correlated. So whether you zoom in or zoom out, you're measuring these differences about individuals, And these individuals are different from each other in how they are moving. And again, we can capture this heterogeneity quite nicely. Problem is, well, when you think about what it's for, especially in a social context, the individual differences are often correlated with other things. So Dima Munoz, in her PhD with us, she was looking at how quickly these stickleback fish would choose the left or right chamber for a food reward and how they learn over time. And we find that the bolder, more exploratory fish are faster to make decisions about their food choices in these contexts. So cognition can be related to personality as well. Not only cognition, but behavioral traits can be correlated with morphological traits or physiological traits. And here, Williams et al. In a PhD with us, she was looking at the red-headed and black-headed, these beautiful Gouldian finches. And what Leah showed was that these black-headed birds were basically bolder and more exploratory than the red-headed morphs. So this is the latency to approach a novel object, and here is the time the birds take to return to feeding after being presented with a false predator. And so the black-headed bird, this morph, this phenotype, is related to a behavioral type as well. So these things aren't mutually exclusive. And when we're looking at behavioral differences, they are often interlinked with other types of heterogeneity. So if we want to try and work out if that heterogeneity is important and how it functions in a group context, it's quite tough to do because there's lots of different types of heterogeneity. And what I'm really interested in is how groups function and how the collective behavior of a system is maybe or maybe not dependent on these individual differences. And so this is a review article by Yules, and also we can start to measure these things, measure these individual traits, and then compose groups of different types of officials, for example, and have a look at how that happens. And this is something that people have been interested for a while. And the way that we most often do it is by trying to understand how these differences change or modify the interaction rules that individuals have with each other. So this is a typical modeling cycle that you have in collective behavior. So you can watch your organisms and you can look at the global patterns, what the whole group is doing, or you can look at the local dynamics and the interactions amongst individuals. Then you can think and imagine what those interaction rules might be and build some models, or you can use your data to inform empirically the rules that you might be interested in. And you can build models like this. This is Daniel Strombaum's model from Journal of Theoretic Biology. And here is just a simple alignment model. So there's very little interaction rules in here except for alignment, and you're getting these patterns of behavior. And then what we do is see how well the model reflects or represents the behavior that you're seeing in the first place. And you can do that cycle as many times as you want. And going back to that idea about heterogeneity, these models tend to assume similar or uniform behavior in all of the agents, but we know individuals behave very differently from each other, just like I've just described. So the challenge is really to try and include this heterogeneity in these models to see how that has, and if it has any impact on the structure of groups and the functioning of groups in different contexts. And this is, like I said, this work's been going on a while, and it's also a kind of difficult area to work in because a lot of the theory is very established, but much of the empirical data supporting this theory is made up. So Jonathan Pruitt did lots of work, published high impact work in this area using social spiders, and it turns out that lots of this data and these results aren't trusted and have been all withdrawn. So we're starting to try and re-look at that and whether this heterogeneity has an impact on group behavior. And this is your timeline off of the website for the master students. And you've heard from Marina Papadopoulou, who was a former postdoc of mine. You've heard from Jens Krause and David Bierbach, his colleague. With those three, Marina's been looking at these mollies, who you've probably heard plenty about from Jens. And so what we've been doing with that to tackle this heterogeneity and organization problem is to use Jens's robo-molly and have the robot fish perform a standard trajectory in a tank, have our real fish behave as it wants with this robot. And what you see is there's differences in the time that these fish spend close to the robot, even though these mollies are genetically identical because they're clonal fish. So this means that all of the other things I was telling you about, well, anything that was genetically innate will be identical amongst these individuals. So we're controlling or kind of keeping constant many of the other things that we might be confounding or interdependent with the behavioral heterogeneity, and we're just measuring the social responsiveness of the fish to the robot. And of course, by using the robot, we are again controlling for the behavior of the other individual in this dyad. And so we're getting a true, or true as we can, measure of the responsiveness of a fish to other fish using a fake conspecific. And then what we do is create groups of fish, shoals of fish, of different types or levels of heterogeneity, so more or less heterogeneous. So we put lots of fish that are similar in their social responsiveness, or fish that are very different in the social responsiveness. And we track them and we see how they're shoaling and what they're doing. And we've got some really exciting results that I think are going to kind of explain that story a little bit more, in how heterogeneity can impact on the collective behavior, at least of shoaling fish species. So that will come out soon, hopefully. And when we're doing these sorts of investigations into how individuals may impact group behavior and how different types of individuals may impact, one of the things that I really like to study is leadership. So that will be when there are one or a few individuals that have a really strong or disproportionate impact on the group behavior. So this would be more like a dictatorship scenario if you want to use kind of layperson language. So this is one or a few choosing versus the complete opposite, which would be everybody gets a say. And actually, theoretical models suggest that democracy should be more widespread in nature because it reduces the what's described as the consensus costs. So that's the cost of an individual performing the same activity or behavior as the others in the group. So the cost is less if you have a democratic decision-making process versus a leadership scenario. And you do see this quite a lot in nature, and it often operates as a type of quorum. So this is where the number of individuals in a group, the number performing a certain behavior or signaling that they're going to perform a behavior, the higher the likelihood of a decision happening. And so where you have to see this sharp increase, this is known as the quorum or the quorum threshold. And this happens quite a lot. So it's when a certain number of individuals do behavior A, the whole group will adopt behavior A. Or when a certain number of individuals say they want to do behavior B, then they will do behavior B. And so, for example, Hallie Walker was a student working with us looking at these African wild dogs. I'll pause that and go back to the start. And these African wild dogs live in Botswana, and they have a strong hierarchical system where they have an alpha pair, a dominant pair, male and female, and they're cooperative breeders. So they have their kin in their group, their offspring. And Hallie was interested in the behaviors before going off and hunting. And so Neil Jordan was there watching these wild dogs. He was running the camp at the time, and he said to us that he thinks he can predict when the wild dogs are going to hunt based on the number of sneezes that they were doing. So he was predicting that here sneezes were acting as a signal that they were ready to go hunting. And this is a little video where you see, I think it's this one here, comes in, sneezes, and then this one will come in and do the same. And so they're just small, or quite big actually, exhalation of air that these dogs are doing prior to these departures. And it turns out that after lots of videoing and traveling around by Hallie to these different packs, it was the case that the more sneezes that you're seeing in this pre-departure period, the higher the likelihood that the dogs were going to move off and start hunting. But we also found this intriguing difference is that this threshold, this quorum here, shifts to the left. So you need less sneezes if you got these, what was called POA 1, 2, and 3. This is the level of dominance of the dogs involved in the rally. So if you've got the most dominant individuals, which is the two dominant male and female, you only need a few sneezes to get the dogs up and hunting. And if they're not involved, you need many more sneezes before the dogs go and hunt. So here we've got a democratic process, but not everybody's vote is equal, if you like. So we have more weight when certain individuals are involved in that group decision process. And that's where we get to leadership. And in fact, across the animal kingdom, we quite often observe leadership in a range of species and contexts. So here we've got these little rock ants that are, you have leaders and followers, teachers and pupils, if you like, where they teach them the direction of the foraging patch or the nest site. We've got waggle dancing bees, which again, a small percentage, like 5% of the colony, will be scouting and informing the rest where to go. So information can be incredibly important to leadership. Here we've got dolphins doing side flops to initiate changes in activity in the pod. And of course, you can think of all the different types of leadership we have in human systems. And one of the study groups that I like to study, you've already seen some baboon work, is the chacma baboons. And they show this type of despotic leadership. So in the baboons that we study, the dominant males in these groups have a bigger influence on each other group members' movement patterns. So when they move, they tend to be followed more often by others. And as a result of that, they have a stronger influence and they have a greater say in where the baboons are foraging and moving. And we see this relationship, these leadership dynamics with three of those different populations that I showed you on that map. We find the same behaviors, these dominant leaders. We haven't looked in the rest. And we even see it when the environment is very different. So in Anna Bracken was studying the Cape Town population, which move into these urban spaces. And when they're in the urban spaces, the baboons are much more spread out. They're less cohesive, have less chance to influence each other's movement. And yet we still see this relationship with dominance, so that the more dominant baboons have greater influence on others' movement dynamics. And so we're trying to understand how you can have the evolution of such leadership dynamics when these theoretical models predict that we should perhaps have the opposite. Okay, that's at 26, not too bad. Okay, so we're thinking now about how that's our overview of our research, right? That's like a kind of highlights reel of the different types of research that we're doing in the group. And then what I want to show you now is some of the ways in that research can develop into different types of research questions. So here we are looking at juvenile sea bass. Sea bass, little tiny, few centimeters long, and they are being raised in different water types. So Ed Pope here at Swansea was doing a big project on ocean acidification. So ocean acidification has been proposed to have lots of different consequences for marine life. There's again lots of papers and lots of controversies about the impact of ocean acidification on behavior and cognition in marine mammals, and especially marine life, and especially fish. And so what we had the opportunity to do here was Ed was raising these fish, and so we started tracking fish raised in standard conditions or near future ocean acidification. So that's increased CO2 and increased temperature, or fish that were raised in normal conditions and then plopped into OA water, and we looked at how they behaved. We could look at their movements and their social dynamics, just like we do with the fundamental research we did. And Matthew Dutel, a PhD student, looked at all of these data, and this was really nice because we had large sample sizes with many individuals because they were doing these ocean acidification trials. And we showed that the sea bass at an individual and a collective level show behavioral resilience to near future ocean acidification. So they don't fundamentally behave any differently in these near future OA conditions. So that's a good thing from the perspective of these sea bass. And from a scientific finding, is a null result, is a no change, is a good thing in this situation. So again, here we're using our tracking systems and our creation of agent-based models to see how the interaction rules of the fish are changing or not in these different, or altering in these different conditions. And some of the other things, again, we're thinking about climate change. These are Welsh mountain sheep that you're looking at from the back here. And here we're putting an accelerometer to the backside of the sheep. And the reason that we did that is because we wanted to know when and where the sheep were urinating. And this was a project with University of Bangor and Rory Wilson here in Swansea. And so what postdoc Lucy Lush did was use these accelerometers to identify urination events, so that the backside of the sheep tips down quite dramatically, and you can capture the frequency by calibrating the amount of urine per second. You can calculate urine volume, and then you can calculate daily urine volume. Why on earth are we doing this? Because you want to know the amount of nitrogen content deposited in the soil and the amount of nitrous oxide that's put out into the environment as a result of these urination events. And so NO2, or laughing gas, is a big greenhouse gas, a contributor to global warming and global change. And so using these tools, we've then got a way of assessing the scale and nature of nitrogen pollution arising from sheep grazed pastures. So people can then use these empirical data to then model the consequences of these urination events and the amount of sheep we're having and what type of land they're using. Another thing that's come, so these two here are really applications of the methods that we've developed for answering behavioral questions put in a more applied context. But something much more straightforward is that when we're studying baboons that are in areas with landscape change, so this is a male Chacma baboon in the Cape Peninsula, stuffing his face with spaghetti bolognese in a restaurant. And so this baboon, through this landscape change, through using human modified environments like he is here, he's gaining all of the calories that he would normally take 9 hours to collect in the day within a few seconds eating the bolognese there. And so what Gail Fellman was doing in her PhD was looking at how these males were moving away from their troop. So here we're looking at the distance from the urban edge of baboons, and the gray is the whole troop. So they tended to be, there's no scale on here, but half a kilometer or so from the urban edge. But these individual squiggly lines are males like this. So this male would be spending most of his time very close to the urban edge. And what they'll do is they'll move into the urban space, find this opportunity for these high calorific foods, and then move back out. And this is problematic from a management perspective. So the Cape Peninsula are interested in trying to mitigate these interactions, and they don't want baboons in the restaurants eating the food, and you don't want to have interactions with people. And so what Gail did was she captured the level of the bottom layer here is calories available to the baboon in its home range. The middle layer is risk. So this is the risk of encountering a threat. That might be a dog, or it might be an electric fence. But more likely, they employ baboon rangers, which move around and actually use paintball guns to keep baboons out of urban spaces. And so by modeling or trying to capture the level of risk and opportunities in these environments, but we were able to tell the management, well, where is it in the space that baboons are using, and why are they using those areas? And by suggesting some changes that offset the costs and the benefits that the baboons are experiencing, the next year, once they followed the recommendations and also made some changes of their own, this was the distributions of the baboons' distances from the urban edge. So no longer are we getting these guys eating these bolognese in the restaurants. We're getting a cohesive troop with these males alongside them. So this is a nice example of understanding how the ways in which animals make decisions as a group or individually, and when we see fission and fusion in these systems, understanding the underlying costs and benefits of making these types of decisions, we can alter those costs and benefits. So here we were putting fences in certain places, and we were changing the behavior of those people with the paintball guns to push in different areas of the home range, and then we can get these different outcomes. Okay, something else that we often do is try and think how the data and the findings that we have from our social systems may be important for different domains, for example, engineering. And here I am in Australia doing a postdoc I did studying merino sheep. The Australians thought it was hilarious that this Welshman would come all the way to Australia to study the sheep, but it was there because these merino sheep were very interesting from the perspective of a bigger project. And what we did, what I had the opportunity to do, was again putting these backpacks on the sheep and the dog. And here is a shepherding dog. And what you see here is all of the sheep in there kind of resting in their field, and we give the dog the signal to go and herd them, go and collect them. And we want the dog to bring the sheep to the bottom corner. And so this is what happens as the dog approaches. You see this collapsing of the sheep. They all form a really tight, cohesive flock. And then the dog sits behind that flock and pushes them forwards towards this target area where we want them down the bottom. And you see there that the dog is kind of weaving left and right to keep the dogs, keep the sheep in a cohesive flock and moving towards that target area. And so we were asking two fundamental questions. Well, main fundamental question here was, how are the red blobs, the sheep, responding to the threat of the dog? And we were looking at selfish herd behavior, where these individuals try and put themselves in the center of the flock, which we were able to show with these data. But when doing so, became really interested in what the dog was doing. So how was the dog actually herding these sheep in these situations? I tried to do it myself. It's really difficult to maintain a speed that keeps these sheep cohesive. They just kind of spread out and fracture really, really simply. And so what we did with Daniel Stromberg, we built a theoretical model, simplest one which we could, to try and recreate the behavior of this system. And it works pretty well. And again, you're seeing that weaving behavior of the blue dot behind them. But actually, we didn't put that in the model at all. What was happening was that the dog sits behind the sheep if they're at a nice certain distance from them, sits behind them, as long as those blobs are together. As soon as the red blobs, as soon as the sheep start to separate and there's gaps in that area, then the sheep dog moves to that flank where there's most gaps. And by moving to the flank, the sheep then respond by attracting to each other, and they get back into that cohesive unit. And so this wiggling, this movement behind the flock emerges as a consequence of a different set of movement rules, which was quite cool. And we had this really simple model then of how you could use really, really simple rules or heuristics to guide things. And so this Daniel here, what he's got here is this little black dot, like the Molly robofish, is just a little robot sat underneath a tank, and it's collecting. There's no water in here. It's collecting these little tokens, and it's collecting these tokens and bringing them home using the shepherding algorithm that we developed from the sheepdog stuff. And you'll see that it's not brilliant. So engineers don't think this is, there we go, pushed one off. So it's engineering perspective, it's not optimal. But given how simple the rule is, we think that you can use this to do things like collecting objects in the environment or herding of different animal systems. So, for example, we might be able to herd bird flocks and fish away from wind farms, maybe herding livestock and predators, which is much more directly related to what we were talking about, or herding bird flocks at airports. There's all these areas where you might want to try and manage or alter the behavioral patterns of collectives, of social systems. And we proposed the one way to do this would be to have an observation drone watching the animals, and then have your herding drone reacting and acting in response to the movements of individuals. And so that's something we proposed, in which Lucia Pedrazzi is looking at her PhD with us to try and work those things out. And it's hard, actually. It's really hard to do. And one of these things, when you're trying to think about first understanding the system, their interaction rules, and then how they're going to respond to a threat, it became apparent that we might be able to use a different system with the sheep and have them be lured by the drone. So what Lucia did as part of her PhD earlier this year was condition the sheep to associate the drone here with feeding. That should be over there, with feeding, so that you're seeing the drone as a positive stimulus, as a positive cue that they have food, and it worked quite well. So what you're seeing here is the red movements of a drone, and then all the colorful ones are the movements of the sheep. When they see the drone, they're following it around. So what Lucia is trying to work out is how long we can keep that association with the food and the drone, how quickly they habituate, how far might you be able to move them, thinking about stuff like ecosystem services and moving sheep around the environment in order to graze different pastures or keep them away from certain areas. And so again, using our knowledge of the social interactions and the reactions of individuals and groups to different stimulus, we can start to think about these more practical applications. Okay, 40. We got a bit of time. So I've gone quite quick, but now we're going to spend some time thinking about where I think collective behavior research might go, or definitely where I'd like to go more in the future. And this is, and I call it pick a system here. So especially when I was earlier in my career, when I was liking to study these different species, so I tend not to be too wedded to a particular system, and often driven by the questions and then develop tools. But often I used to get that statement, well, if I'm putting in a fellowship application, well, why doesn't he pick a system, pick a model system and stick with it? And part of the reason was that I was always really interested in the differences and similarities we see across the animal kingdom in terms of how their patterns of collective behavior emerge, right? So this was the start of the talk. These shoal of fish just in their milling state, the kind of basic state that you see. So these collective movements that we see across lots and lots of different groups, and if we just pick vertebrates and think about your caricature of fish schools, bird flocks, mammal groups, there's lots of things which you think of which are quite similar about these movement patterns, and there's also things that you could bring up as obvious differences. And so what I really want to try and do, or what I think we as a field can start doing, is we focused a lot on mechanisms. So that's how the interactions between individuals produce global patterns of behavior, mostly from self-organized perspectives, where you're having these simple local rules of interaction producing these global patterns. But we've been able to tackle less questions about the origins and evolution of these types of behaviors, these collective behaviors. How does it evolve? How many times does it evolve? How similar are the functions of these different behaviors? Are anti-predator collective behaviors similar to migratory collective behaviors? All of these types of questions are pretty open in the field. And the reason I think it's interesting to start to do that now is even in our group, we've got lots of different time series data now for multiple individuals. And as a field, we've definitely in a position where we're getting lots of these data. So XYTID, so this is positional data through time for many individuals. And so this will look something like this. So these blobs are individuals moving in time and space. And actually, this is some of our baboon data. So these are baboons moving across their environment, and you're seeing them move in these kind of, these are called progressions. And this is something that Marco Felle studied recently. And what he did, we looked at, these are days that our baboons have our collars on. So I think there's about 30 or 40 days here, and then start of the day, end of the day. And the color represents the level of coordination amongst individuals in this troop. And so when it's brighter color, they are more polarized, which is more aligned, and they're moving faster. And so these little snippets represent coordinated motion. These represent collective behavior. And so this, in baboons, are periods of collective motion, which you would see 98% of the time in pigeons that are flying around in the sky, right? So this is a way that we can identify patterns of collective motion or collective or coordinated behavior in time series data for multiple individuals. And Marina Papadopoulou, that's what she did. We've created a swarmverse package now. So any raw data from a GPS or camera tracking or anything which is relatively high resolution data, you can take that data set, and then use thresholds to identify these periods of collective motion. So these events, these events of collective motion. And you can do this with any data. There's a bit of, user knowledge required to select speed thresholds and things to identify the coordinated motion, but there's also some bunch of sensitivity analysis in this package as well. And what happens is then we can extract events of collective motion and then calculate a bunch of stuff about each event. So you can look at things like the nearest neighbor bearing angle, the nearest neighbor distance, or the shape of the collective, or the global polarization. There's lots of things you can calculate. And so this package calculates these metrics about each event that you've identified. And then it can plot and compare in different ways the events that you see. And so, for example, you might plot different species. These colors might represent different species, or they might represent the same group in different environments, or different tanks, or different seasons, whatever you might want it to be. And so you can start to compare what Marina calls swarm space, compare events of collective motion within and across species. And I think this is going to be really, really transformative, I think, because we can start to play around with this. And this is a case study which Marina led, where we used our stickleback fish data, or Marina's worked on pigeons. We used some of that data, goats, which we've done before, and baboons. And so here is an example of one collective motion for fish, one collective motion for birds, goats, and baboons. And so we extract these events for all of these different species. What we end up with is that we can plot each event representing each species. So pigeons in red, for example, that's the events that we've got. Goats, baboons. We have more baboon events because we have more data over a long time period. And perhaps unexpectedly, you can see that when we collect all these metrics and we put them and we use data reduction techniques to get them into this 3D space, that the baboons are different to the sticklebacks and are different to the pigeons. But it's quite interesting that you get the goats, for example, their events are quite often very similar to the baboons, and they're quite often very similar to the fish, and even stretching out maybe a little bit towards the pigeons. So the data, the event for the goats often look exactly like a baboon data set. And so that's the sort of thing we're interested in, these different events across species, because then what we can do is going back to these individual interaction rules. And here we're seeing the positions of the neighbors from a focal individual, the first, second, third, and fourth neighbor. And the hotter the color, the more likely you are to see a neighbor in those positions. So fish forwards and just behind, pigeons side to side. Makes more sense if you're traveling constantly in the air. You don't want to bump into them straight ahead. Whereas your goats and the baboons are much more front back. And that you saw in that GIF, that animation where the baboons were forming this line formation. So we can look at the way they're interacting with each other, and then we can build those models again. We build those agent-based models, and we put that agent-based model through Swarmverse. So we extract motion events from the model, and then we plot it in the exact same space. So that's what Marina did here. She built a pigeon agent-based model, ran it through, calculated the metrics, plotted it over the space, and very nicely and thankfully, the model is sitting right exactly where we see the original empirical data. And so this also provides a neat way of closing that cycle where we're trying to say, well, how similar is the model that we've created to the real observations that we've made? And this is quite nice because we're not taking the interaction rules from this data, but we can put it back in there and see how it fits. So we can start looking at the mechanisms in a lot more detail. And we're still working on this. We just recently found that we didn't get funding to do a big project with this, where we've got Marina, Will Allen at Swansea, and Rob Heathcote, who's doing a lot of the fish biology in Oxford. We've got 33 project partners on board from across 12 countries, where we're getting positional time series data for lots of individuals. So we're getting hundreds of data sets for different species to try and have a look at both how and why we see these patterns of collective behavior across. For example, this is a fish phylogeny, and the blue dots represent fish species for which we have data or will have data. And we can start to look at the behavior in this swarm space, build mechanistic models of how they're doing it, and then map this to function. What's the function of these different patterns of behavior? Are the rules similar for these different functions? We can really start to talk about then not only the mechanisms, but the evolution of these different collective behaviors that we see. And then the really cool part about when you're starting to bring data sets together, here, for example, are those 4 baboon data sets, which I started the talk with. And so we run the baboon data for these 4 sites through the Swarmverse, get the events, and look how similar they are. So all of these colors here are Chacma baboons. And this one here, this green part here, is data from the University of Constance and Meg Crowfoot and Ian Cousin and Ari and all the people in Germany there with nearby you. This is their data from a science paper with olive baboons. So we can put that in there and see how similar these events are. And it's often contrasted that the science paper that they had says that the olive baboons have a very democratic leadership style. So there's actually no leaders, which is contrary to what my work has shown with the Chacma baboons. And so what we can do, remember, because each of these dots represent an event, so each of these is a collective motion event, we can almost time travel and think about the start of each of these dots. So this is when they started moving. So if they were stationary and then started moving, and that represents one of these events, we can go back in time and think about how they transitioned from stationary to coordinated and how it worked. And we started doing that with these data with the olive baboons and the Chacma baboons. And this is a simple metric of polarization times speed. So they're not moving and then they're moving. And you see these similar patterns that we're seeing in the 2 species. And actually, when we're studying the same thing in the same way, we might not get quite the same contrast of results that have been portrayed in the literature, perhaps not by us, but by others. So that's a really exciting part as well. We can go back to these data sets and start comparing apples with apples instead of different contexts and types of behaviors. So that's a really overview of everything that we're doing. So the fundamental research and the methods that we're doing to understand everything from the SHOAL, sociality, heterogeneity, organization, and leadership, how that then leads to some exciting projects which perhaps we didn't foresee, thinking about societal and environmental challenges. And then doing all this stuff with different systems and different species, I think really provides a really unique opportunity to start doing comparative research across collective behavior, rather than our focused on certain species and certain sometimes quite unique contexts, which might not perhaps be particularly ecologically relevant. And so that's where I'll end, and I'll stop there, and then we can take questions. Thank you.